And then Judy, I will turn it over to you to start.
Good afternoon, members of the JCC, and welcome to our panelists. I'm Judy Gasson, and I was director of the Johnson Cancer Center and president of the Cancer Center Foundation from 1995 to 2015. Our current director, Mike Titel, couldn't be with us today, so I'm standing in for him and welcoming all of you to this discussion of our new partnership with AUTOBAHN. In order to give you a little bit more information about that partnership, I'd like to introduce my colleague, Mark Wisniewski, who's the Senior Director for Biopharma at the Technology Development Group. Mark?
Thank you, Judy. And I'm excited to be here today and welcome our AUTOBAHN, Evotec, and Samsara colleagues to really provide an overview of this novel program that we signed in June 2020, or 6 months ago, I guess, time flies. And really the essence of this is that with the AUTOBAHN focus on early stage research, we're here to enable and facilitate startups that really have PI involvement. And I think what's really powerful about this is their recognition that PI engagement is critical for early stage startup success. And with their focus being earlier than what your typical venture capital or an angel investor would be, you have an opportunity to really come out of the gates on the right foot in terms of what the program focus should be. You have the PI involvement. And what we've done at TDG is simply execute standardized agreements between options and licensing. So we're out of the way, so the focus is really with the AUTOBAHN team to say, who are the best PIs, the best programs, and how do we ensure that we get these going efficiently without having a lead negotiation time. On that, I'd like to turn it over to Tom Novak, who's the CSO of AUTOBAHN, and welcome everyone.
Thanks, Mark. Thanks, Judy. I very much appreciate it. And I welcome all the UCLA faculty members listening in and appreciate your interest in learning more about AUTOBAHN. Marvy, if we have the first slide. Thank you. So as Mark said, we signed a deal. AUTOBAHN Labs is a new incubator that was founded in 2019 as an investment between Samsara Biocapital, a VC firm, a biotech VC firm based in Palo Alto, California, Evotec, the large global German-based CRO, and KCK Group, a single family office that's investing in tech and biotech and a number of other areas. And it was formed with this sense that there is a lot of exciting research being done in academia, but at an early stage, it lacks the funding needed to really accelerate it towards the clinic. So our focus is on therapeutics, and I'll, as we go through the slides, I'll give you additional information as to what's in scope, what's out of scope, how we go about evaluating programs, and what we're looking for from faculty at UCLA. We're very excited about this. You're our first institution that we've signed up, and it's been an incredible experience since June to have the number of conversations we've had, the exciting projects that we've heard about, but we want to expand it even more and make it available and understandable to the bulk of the faculty at UCLA. So if you could go to the next slide. Yeah, and so as I said, our goal is to partner with universities to bring drug discovery and development opportunities to campus. Typically, universities are a great source of innovation, but in terms of drug development and discovery, they often they aren't designed to have those capabilities. They're designed, and the focus on the faculty is on novel science, groundbreaking science, but the application and the conversion of those discoveries into a therapeutic is just not the focus of the university. And that's what pharmaceutical companies, biotech, CROs are there to do. And so what we want to do is be that partner for your exciting early stage research and help move that from an interesting biological phenomena into a potential therapeutic. And so the next slide sort of illustrates this, this point. There is a lot of exciting work coming out of academia. Marvin, can we have the next slide, please? There we go, thanks. And here you can see, smack dab in the middle is Xtandi, that was discovered by Mike Jung and Charles Sawyers at UCLA. There are a fair number of recent Nobel Prize winners and National Academy members listed here. But it really just goes to show that a lot of amazing research and groundbreaking research comes out of academia. But it's the exploitation of that, the commercialization of that, the conversion of that to a therapeutic is where it sometimes can stumble. And that's what AUTOBAHN Labs has been built to try to facilitate, to move these ideas from an early stage of discovery into a potential therapeutic. So if we go to the next slide, how do we differ? There are other incubators out there. There are a lot of search and evaluation efforts going on from VC firms, from pharma and biotech looking for new projects. But what we typically find is that they're looking for assets that are perhaps 12 to 18 months from the clinic. So you can, which is fine if you're at that stage. But we believe that there is a lot of opportunity, untapped opportunity, that is much earlier than that. Could be 4 to 5 years from the clinic. And that's what we're set up to do. These programs are seen as risky. In fact, they're too risky for one of our founders, Samsara. And one of the goals we have is to find this early stage research and de-risk it so that it is at a suitable point where a traditional and typical VC firm would make an investment. So we want to help bridge the gap from this very early stage research to the more IND enabling. And if you go to the next slide, Marvi, it really just shows you where AUTOBAHN's sweet spot is. And so it's to go from this 4 to 5 years from the clinic. We're willing to invest up to $5 million and 3 to 4 years to get to this preclinical development candidate. Then there's another usually year or so to get you into the clinic after you do your IND enabling studies. So we're looking for programs that could be 4 to 5 years from the clinic. You may not even have filed an invention record yet, or filed a patent on these discoveries. But those are the things that we're interested in learning about, because we believe those are ideas that deserve some investment, and we can provide that with the team of drug hunters and the expertise that we bring and capabilities that we bring to this endeavor. So the next slide, Marvin, as I said earlier, we're willing to invest up to $5 million to translate this work into a potential therapeutic. Our model is the majority of the work we do will be done at Evotec because they have the expertise and the capabilities. And because often these are not skills, but they're efforts that are often lacking at a university, to have a big drug metabolism and pharmacokinetics group, to have a big medicinal chemistry group, just churning out analogs is really something that is best done at a drug development organization. So what we do is, we'll assist in the target ID and validation. That may be where you're starting. You may have a phenotypic screen where you have some interesting biology, but don't know the target. Maybe you have identified the target, but don't have good chemical starting matter. We can work with you there, either setting up a target-based screen or helping to do target identification on phenotypic screens. That's something we've been hearing a lot about from UCLA. I know you have an excellent screening core there under Robert Damiso, and we've been very impressed with the data that we've seen come out of that core group. Obviously, once you have a target, you enter into this hit identification phase where you're either running a high throughput screen or you're doing hit to lead confirmation. You're really trying to figure out what's the starting point for a medicinal chemistry opportunity. Then going into lead optimization to actually improve the drug-like characteristics of a molecule, specificity, potency, half-life, avoiding off-target effects on CYPs and improving oral bioavailability, all the work that needs to be done to convert a molecule into a clinical candidate, and then finishing up with some preclinical toxicity. If we get to this stage, then we stand up a NewCo, and that's something that would be done by Samsara. So that NewCo would then handle the IND enabling and subsequent clinical development. In effect, this project would then go out of AUTOBAHN's portfolio and into Samsara's portfolio. So as we said, up to 5 million, 3 to 4 years to get through this phase. It can be faster. And UCLA and the PIs receive equity as founders as their return on this agreement. If we fail to hit a go-no-go milestone and terminate a program before we get to this NewCo stage, all of the data that we've generated goes back to the PI, and they're free to use it as they wish. So we think, again, there's a win-win for everyone in this opportunity. So next slide, Marty. So what do we bring to the table, and why should you be interested in working with us? So we bring this intellectual capital. We have a team, I'll show you on the next slide, of very experienced drug hunters, up to some folks as many as 40 years in the pharmaceutical industry, have seen a lot of problems, have solved a lot of problems, and can really offer an incredible insight into the best way to take an early stage discovery into the clinic. And that's something that we think is of incredible value. Physical capital, we work with Evotec. Evotec is one of the premier CROs in the world. Every major pharmaceutical company works with them. They have a proven track record of accelerating research. Iva Toudjarska, my colleague, will talk about this in a few slides. But we bring their physical plant, their capabilities, their instrumentation, but it's also their intellectual capital as well. And then lastly, we bring this financial capital. We have visionary, patient investors. We are, not only are we willing to invest up to $5 million to get this program code, this early stage project, to a new company phase, but we also have then the follow-on funding. So if you go out and raise your own money, get some angel funding, you still, once you've spent that, or at some point when you're running low, you need to go out and raise another round. And that's a lot of work. And every time you need to go out and do that, it can be a real delay in the productivity and the advancement of your program. We have a built-in mechanism for raising that next round of funding. And again, Samsara, Donnie will talk about this in a bit, where it's not a typical firm. It's has a long-term investment thesis. And that's what is allowing them to invest in these early stage programs. It's not a 10-year fund that has to wrap up and return money to the investors in a few years. They can stay much longer, invest much longer, make multiple investments in a company that they find attractive. So these are 3 areas that we think really distinguish us from some other efforts where maybe they're coming in and licensing something, you never see them again. We want to work with the PIs. If it's early stage science, it requires your input, because you know this field better than anybody, certainly better than we do. We would not come and license IP without the PI's involvement. We want you to be involved in helping us develop the project plan. We want you involved in team meetings as we discuss data in real time. And if we're successful and lucky enough to stand up a new company at the end, we would hope that you would come along as a member of the scientific advisory board. We want PI involvement. We're not interested in just coming in and seeing what's for sale and taking licenses and doing this on our own. So I think that, again, is a distinguishing feature of AUTOBAHN. Next slide just shows my team. It's incredibly talented group of individuals from Michelle, who's our SVP of operations, and Yushi, our project manager. We have a great board. We have advisors who work for Samsara. We get a couple of days a week in various areas, pharmacology, medicinal chemistry. We have a scientific advisory board, as I said, with between 20 and 40 years experience in drug discovery. And then, board members like, Srini, who's the managing director of Samsara, Krishna for KCK, Thomas Honka, who represents Evotec. So almost all of these folks have PhDs. Most have worked in drug development and discovery for a number of years, and this gives you a great resource to bounce ideas off of, to help put together project teams, and most importantly, to solve problems that arise. The drug discovery is fraught with them. One never advances a program without some unexpected pitfalls, and it's good to have some people who have seen a lot before to help work our way through those. So next slide, Marty.
Tom, it actually looks like there's a question, if you want to take that.
Yeah, we were going to wait till the end, Donnie, to answer
Okay.
questions. So if that's okay, we'll do that. But I do have the answer for that question. Why don't we just wait, and then we'll take them all at the end. And again, this is what we do. When we evaluate a proposal that comes to us, we do look at the commercial landscape. What is the current standard of care? What are the unmet medical needs with the standards? What compounds are in development? Do they promise to address some of those unmet needs? We make some guesses as to where we think the market will be in 15 years, 12 to 15 years, when a new drug would hit the market. What are the development challenges, regulatory and clinical hurdles that we might have to face? And we put together, so we look at all of these opportunities, and a fair bit of it is, as Michelle will say, looking around corners, looking through walls. We have to make some guesses here, but what we're looking for are novel therapeutics, so in areas that aren't too crowded with existing drugs, especially those that are generic, and we're not really scared away by the fact that a lot of these discoveries may be for very, very early stage. That's what we think we have. There's some value to be had in investing early and partnering early with faculty here. So with that, we do this in conjunction. Evotec's in the middle here for a reason. When we start to put together a project team, if we find a program that we like, we invite the PI to work with us and Evotec scientists to put together a project plan. But generally, this is driven first by Evotec based on the input we have on the project, the target, the indication. We then involve the PI, the AUTOBAHN team, and we really start to nail down this project plan. And it includes, again, evaluation of the market and the opportunity, the developability, also the specific steps. What are we doing? High throughput screening, then we're doing hit to lead. What are the go, no go criteria? Are we doing target identification first? We lay all of this out before we go to our board for approval. So with that, I'm going to hand it over to my colleague, Iva, who is a SVP or EVP at Evotec. She's a member of the board at Samsara. She's involved in all of our discussions on evaluating programs, and she's our conduit into the Evotec scientific community and as an invaluable part of our team. So Iva, I'll hand it over to you and I'll go on mute for a little bit.
Thanks a lot, Tom. And I'd like to thank Judy, Mark, and Murray for hosting us and all of you for joining today. Tom did introduce Evotec very briefly, and even though historically the narrative has been a CRO, we prefer a global R&D partner. And I hope through the next couple of slides and 10 minutes or so, I can convince you of that really major shift in how Evotec operates, including our partnership with AUTOBAHN and UCLA. And so very briefly, as an introduction, Evotec has been around for 30 years. And we have at this point about 3,400 scientists distributed globally throughout. And our core business is within research, discovery, and development. Anything from target ID and validation all the way to IND, including a CMC capabilities all the way to market. You can see here sort of a little snapshot of what we can do, and I'll walk you through some sort of key points that might be relevant for all of you on the call, and definitely welcome questions and comments afterwards. And so you can see within the target ID and validation, we have state-of-the-art methodologies that we're employing. Traditionally, we have been known as a small molecule powerhouse. However, in the last 3 to 4 years, through both organic growth and acquisition, we've grown significantly and acquired Just Biologics in Seattle and have state-of-the-art antibody generation and manufacturing capabilities. More recently, gene therapy and RNA capabilities have augmented our earlier stage gene therapy in addition to our GMP manufacturing capabilities. And so and more recently, even cell therapy as well. So we're truly modality agnostic. On the chemistry side, I know some of you are eager to know who can help with medicinal chemistry and molecule augmenting molecules. We have 700 chemists at Evotec, so really very significant resources to partner with you on advancing molecules forward. Very strong and proprietary research informatics and proteomics and metabolomics platform covering multiple analytes, multiple tissues, multiple ways to get there and find that valuable information, including building our own patient-derived biobanks. And on the biology, in vitro and in vivo side, very extensive capabilities, including 3D tissue cultures, organoids, patient-derived samples. We have a partnership with Oncopol in Toulouse, which is, we're fully integrated with their up to phase 1 oncology capabilities. And also there on the bottom, Indigo, which is our IND preparedness platform to take products all the way to patients. We partner for clinical development. And so in addition to all of these, so very technical capabilities, we have very broad therapeutic focus and interest, anything from infectious diseases, lung disease, kidney disease, fibrosis, oncology, and everything in between. And as I did mention, platform agnostic. If you can advance to the next slide, please. So Autobahn really means the proverbial German highway that has no speed limit. And that's the goal of the partnership, to be able to take novel academic science and accelerate its discovery and development towards therapeutics that can reach patients. This is not a very new concept for Evotec. As I mentioned, not away from sort of the CRO model, we have initiated a number of different partnerships we call bridges. And bridges really serve that valley of debt where for academic model to transition into a venture funding or a pharma funding, there's just not enough either industry knowledge or funding or capabilities to get there, but you still need data to get that funding. So that valley of debt, we create a bridge where we come as an operations partner. And we have several global partnerships that are operating, one with University of Oxford, another one with 15 universities in Toronto. And I'm more than happy to take questions later or offline to answer how do we operate there. And the reason for the need to have structures like Autobahn is presented on this slide. There's a number of research that has found that oftentimes academic science, it's very hard to reproduce in industry setting. This is my personal experience too, running search and evaluation within Alnylam Pharmaceuticals for almost 4 years. The reason for that is quite simple, just lack of industry, robust and scalable platform. And so what we really do is we partner with academics, as Tom already articulated, evaluate the science and accelerate the science of Evotec's industry platform. How do we do that? If we can go to the next slide, please. Thank you. So bridges really symbolizes that bridge that spans over the Valley of Death, but actually it's an acronym that stands for Biomedical Research Innovation Development Generation Efficiency. So we have, as Tom explained, what the process is. We have collaborative evaluation process together with AUTOBAHN's advisors and the PIs and Evotec scientists, both therapeutic area experts as well as technical experts. We look into each project, and our considerations are quite simple. It's scientific novelty, developability, which is really what do we need to get done to get to generating a molecule, animal proof of concept, and then even human proof of concept, and how long and how much we'll need to invest to get there. And third important criteria is commercial viability. Is this the right patient segment? How do we need to position compared to standard of care and competitors? So what you can see here on the left-hand side, these are examples from some of our other bridges. And currently we have over 45 projects that we run jointly at University of Oxford and Toronto. Our time to evaluation is about 2 months before we can make a decision. And as part of that process, PI is extremely engaged. This is the same model we're applying here for AUTOBAHN. And we always provide feedback to you on what are some of our considerations with regards to the particular project and its development. The next stage really is time to first experiment. And the graph here is a little bit different. So we still have, that's 8 weeks, which is still 2 months. And so Evotec as the operating partner in this is the team is very well positioned after we have created the work plan that Tom described jointly with the PI to now execute the project in a very short and efficient manner. And 3,400 scientists, there is knowledge and there's capability to accelerate the projects. And we typically take projects that are about a year to 2 to a value inflection point. Of course, AUTOBAHN here, the goal being to get to IND stage with a $5 million investment. We have a longer path and time to get there. Next slide, please. This is a very high level track of success for Evotec. And this is where, we diverge very significantly from a CRO and much more of a global partner. We currently co-own about 100 different assets in different stages of development. And for the past 5 years, we have started and are part owner of 14 different companies. And again, through our expertise and our platform are enabling drug discovery and development towards patients with significantly unmet medical need. I welcome any questions afterwards. I'll stop here and pass the mic to my colleague, Donnie McCarthy.
Great, thanks, Eva. Yeah, let's go to the next slide. Okay, so yeah, really great to be here today. Nice to connect with you all sort of virtually here. I've been charged with sort of giving some perspective on the investment side of AUTOBAHN, and actually come to it with a little bit of a background both from KCK and Samsara. And so maybe just briefly about myself, and as it pertains to Samsara, we we are a science first fund, essentially. Every one of us is technically trained. As Tom mentioned, we'd like to take a very long-term view on investments. So I think a couple of sort of very unique things about Samsara as a fund. It's a $400 million fund, started about 3 years ago in 2017. It was actually designed in a way and actually set up from a corporate structure perspective in a way that allows us to have, a 10, 15, perhaps even a 20-year horizon on our investments, which is, as many of you may know, very atypical of venture funds, which often have a sort of 10-year lifetime and 10-year horizon. And the rationale for that was actually quite simple. In biotech, it takes a long time to really create a lot of value, and we wanted to do investments early, and actually that's part of the reason why AUTOBAHN Labs was such an interesting concept to us from the beginning. It allows us to get into this sort of early stage, high quality academic science, and then allow sort of the facilitation of that into preclinical candidates, and then onward into clinical candidates, and into the later clinical stage drug development, and ultimately to commercialization, which can take 20 years, right? And there's a lot of examples of how that's actually played out. And original funds were often structured such that you had to pay back your LPs within 10-year timeframe. And so there's somewhat of a conflict of interest, so to speak, with how the investment team, like myself and my colleagues, sort of view an investment versus how our LPs are asking for their money back, right? And so to avoid that sort of conflict, the fund was structured in a way where we don't have that obligation to pay back investors at the same time frame, and we can actually look 15, 20 years down the road. And I think it's a really important distinction that Samsara has. And so besides that, we, as I mentioned, we're all technically trained, PhDs, MDs. We really care about the science. I mean, that's sort of, when we look into any deal, whether it be early, mid, or late stage, we start with the science. For us, it really has to be exciting, compelling science that really supports sort of the rationale for whatever drug intervention that we are pursuing. We tend to be focused exclusively on drugs, and that's sort of also within the AUTOBAHN guise, is we want to invest in and support next generation therapeutics. But beyond that, we tend to focus really exclusively on the science and how that supports sort of the ultimate rationale. And so that's Samsara, very briefly. The other investment arm here is KCK, and the KCK, as Tom mentioned, is a family office, so a single family office that, as he also mentioned, invests across industries, but has had a long-standing interest in biopharma and actually in medical devices. And so they have actually a fairly sophisticated team, as family offices go, for investing in very niche opportunities like in biotech or in biopharma. And what also is sort of well suited for this particular opportunity is that they don't have LPs, right? They are the money. And so because they are the money, they can also have this sort of long-term vision, much like Samsara, in that we can still take that 5, 10, 15, perhaps even 20-year horizon for our investments. And so when we were conceiving of the idea of AUTOBAHN Labs, it made a lot of sense to sort of partner up with them, because we can take that long-term horizon and we can start to invest money in these early, discovery stage programs, and then see them out, the next 4 years to a preclinical candidate, perhaps the next 4 years into the clinic, and then probably another 4 years, perhaps, or more, to get it into commercial stage. And that's something that we sort of relied on and made a lot of sense from a sort of investment perspective. So that's the two investment arms here. Obviously, Iva just went through Evotec, so I won't spend any time on that. We'll be happy to take any questions from any of you here at the end. And then I guess in the meantime, I think we have a few more slides that Tom will take over.
Thanks, Donnie. Appreciate it. And thanks, Eva. So as you've heard from Eva and Donnie and myself earlier, AUTOBAHN Labs is focused on developing therapeutics. We're agnostic as to the disease area, and we're also agnostic as to modality. So as Eva said, Evotec had been known mostly for their small molecule expertise, but they have incredible capabilities in developing biologics, cell and gene therapy. They do a large number of iPSC-based phenotypic screens. So they have an excellent group there. They've just opened a facility, I believe in Austria, to do gene therapy and oligonucleotide drugs as well. So again, positioned to cover all areas of modalities, the therapeutic modalities, and the programs can really come from most anywhere. They could be, they could arise from genetic or genomic studies of disease versus normal individuals. They could be phenotypic and target-based screens. We've been impressed with, as I said earlier, the number of phenotypic screens that we've seen run at UCLA, and those don't scare us at all. We can do target identification work quite easily with the group at Evotec. And you may even have a development candidate. You may have something that's further along than what we're looking for, but don't hesitate to contact us if you're looking for some support to move it forward. If you've had trouble raising money or you just need some expertise as to what the next steps are, we'd be interested in those as well. So just as though some things are in scope, there are some that are out of scope. We're not interested in developing reagents or tools or assays unless they're required, obviously, for a drug discovery program. We're not interested in biomarkers unless it's part of a therapeutics program, then we would support that. And then reformulating, repurposing drugs, healthcare IT or devices are out of scope for us. And that again is just playing to the strength of the team that we have and our focus is on therapeutics. So something's out of scope, but there's a large area that's in scope. Next slide, Marvi.
Can I add just one point, Tom?
Sure, please.
And one of our big goals is to build relationship with faculty and with researchers. And so even if you do not think that your program's, there yet, do reach out, tell us, what you're working on. We'll be happy to provide opinion on what we think is the best next step for your program, or how to position your platform. What would be some great data to generate through grants or other means if you need that feedback. So we're happy to engage in this way and help you generate the data when we will be able to take the program on.
Excellent point, Eva. If you've been told either by TDG or by an investor that your project's too early, don't hesitate to come to us, because that is our focus. If they're too early for them, they may be right up our wheelhouse. So as Eva said, yeah, conversations are important with us. And we do believe in building this relationship with the campus. And so our process, we've touched on some of these earlier. Let us know if you have an idea. We've been reaching out to PIs just based on what we've read, what we've seen in the literature, people we know. We've talked to TDG, who have made some introductions, some of the business development officers. But we invite you to let us know as well. And there's an email address at the end of the presentation. If you don't get it down, don't worry. Ask Judy, Mark, or anyone at TDG, and they'll put you in touch with us. We're not contemplating at this point an RFP process. That may happen in the future. But if you have an idea or a project and you're interested in talking to us, please just drop me an email. What we then typically do is have a short phone conversation, 30 to 45 minutes, talking about what you're working on, what the indication is, where the project stands. We'll often, if we make a decision that it sounds interesting to us and within, and again, within our areas of interest, we think has some legs, we'll ask you for a slide deck if you have one. We'll then review that with our science team with an AUTOBAHN. That typically results in more questions back to the PI. We'll follow up to get more information. We have a slate of key opinion leaders and consultants that we rely on if there are questions outside of our area of expertise. We start doing assessments of the commercial opportunity as well. And then we put together a work plan. We do have multiple approval steps. The science team, our SAB, they're not gating. The SAB is just to let us know if we're missing anything. And same with Samsara, since they're going to lead the A round. If we stand up a new co, it's important that it's something that they're aware of, is potentially coming down the pike. Also something that they feel some comfort in investing in. There's some areas that they invest in less often than others, and so we want to make sure they're comfortable with the things that we're proposing. We then have a selection committee, which consists of Amir and Mark at UCLA, and myself, and Michelle at AUTOBAHN. and we approve this program. We then take the budget for the work plan that we have, and we present both of those to our board of directors for final approval. But during this process in the middle here, this project review and putting the work plan together, that's where we're in regular contact with the PI. As I said, we want your input. You know a lot about the target, and we want your input on what's been done. May not all be published, may not be in your slide deck. And we want you to understand why we're proposing to do the things we're doing, because we think that's another advantage of Autobahn, is helps educate faculty who may not have been involved in a drug discovery program as to what the steps are, the necessary steps. It's very different to get a drug into the clinic than it is to get a paper accepted or a grant approved. You know, those are important metrics, but they're different goals and different requirements than this regulatory process that we're going through. So we think it's very useful to have that back and forth. We think you'll then become more adept at seeing where you may have some commercial opportunity. We've talked to some faculty who really haven't thought about it until we've raised the issue with them. So that's something that we're hoping we can provide to the campus as well. And then assuming we approve this program, you know, then Evotec and the Autobahn team, we oversee this program. We have multiple stages along the way with go, no-go criteria that we have to hit in order to continue the program. So Marvin, if you go to the next slide. Yeah, and so we've talked about some of this before, you know, how we help to choose programs. You know, obviously, we want something that has some novelty in the science, some idea that we can drug this pathway and do it relatively selectively with minimal side effects. So, you know, how well the target or the pathway has been validated. We know in some cases that won't be very well validated, and we're okay with that because it's an early discovery. And then, you know, you're always trying to make some assessment of the probability of technical success. Very hard to do, but, you know, we have to think about this every step of the way. Where are things potentially going to fail? Since drug discovery is a high failure rate industry, most of the things we work on never get into the clinic, let alone onto the market. And so we try to see where there might be challenges in the future. We look at the development potential. You know, so again, is there a well-trod regulatory pathway to market, or are we blazing our own trail? I think people in the cell and gene therapy areas are learning that. They're learning along with the FDA as to what is required in terms of preclinical safety and efficacy studies to get into the clinic and then get approval. Small molecules, a lot easier. Biologics, again, much more of a well-trod pathway, and that reduces risk. We have some idea of what the FDA and the EU, EMA is expecting. So very important to us. Is there a fast path to a clinical proof of concept, or at least to show that we're hitting our target? That's important as well. You know, we have long programs that are going to take many, many years, thousands of patients, and a long follow-up time to see efficacy. Those are more challenging for us, and those would be probably rated lower. And then we put together always what we call a target product profile, which is what are the attributes that this compound would have to have, or this biologic would have to have, in order to be commercially and clinically competitive. And then again, you know, the impact. What is the opportunity for impact? And again, you know, IP is important. We're always asking about IP early on. We're willing to pay to file if it hasn't been filed. I know universities never have enough money to file patents on all of the discoveries their faculty make. We're willing to reimburse for that. You know, do we think we can partner this at some stage down the road? Is how differentiated is it? What's the competitive landscape in which we'd launch this? So these are the sorts of things that we do when we're making a decision. And then next slide, Marty. And you know, one of the things we'll ask for you is, you know, do you have, you know, what data do you have? Could be in vivo, ex vivo, in vitro, really, you know, it doesn't care. We just want to know, what have you done so far? Obviously, if you've done some of this in humans or in patients versus control samples, gives us a little more confidence, but it doesn't have to be. It's just one of the things that we weigh. You know, what do we think the likely indication is? And I'll tell you, if you come to me and say, I have this for pancreatic cancer, you know, we may say that's a really hard indication. Are there other cancers in which it may be useful for? And, you know, a lot of our KOLs tell us Don't worry about the indication. Get the drug first. Get the molecule. We'll worry about the indication later. I don't want to be that flip. It's not that simple. You do need to have some idea of where you're going to go forward, but there can be some flexibility there. We're often surprised when we get into the clinic, but obviously we should have some idea of what are you proposing this for? What's the data that support that proposed indication, that patient population? And then again, is it convertible to to therapeutic development? If it's a cell surface receptor, a lot of ways we can attack that. Secreted protein, a lot of ways we can attack that. Intracellular, again, a lot of ways we can attack it, but some targets are just more difficult than others. And so we weigh all of these as we decide if this is a good fit for us. And then the next slide, I think it's just a very cryptic case study. So there's a program that we're very close to endorsing. We haven't yet funded it yet, but I think it's just a matter of time for a GPCR that's been shown to be upregulated in a particular tumor type. And then there's a lot of work in genetically modified mouse models showing that if you knock out this GPCR, you see a great reduction in tumor number in animals with tumors, tumor size, tumor volume. In vitro, it can induce a certain cell type to secrete a number of immunosuppressive and pro-fibrotic factors. Knocking it down shows reversal of that effect if you take cells that are already expressing that gene. And a lot of human clinical data from the Cancer Genome Atlas showing that tumors of this type show greatly elevated expression of this target. So these are just some of the data that help us get some comfort that this is a good target going forward. It's a GPCR, we know they're druggable. The majority of drugs on the market target GPCR, so that's not a challenge for us. It's novel, no one's worked on it. So again, we don't have any competition. Also means we have to light our way a bit because there's not been a lot of work done on this, but that's okay. We're fine with that. The strengths of this proposal outweigh any of the challenges of which all programs will have some. So next slide, Marvy. And so again, just summarizing what we've talked about before, we have deep scientific, clinical, and commercial support, knowledge, expertise, so we can provide that diligence for projects. One of the things, as Eva said, because academic published results aren't always reproducible, one of the first things we do is replicate the key findings that got us excited. We think that's very important. It should be doable in another setting. If it's very specific to one lab, it either is a very tricky assay or it may not be as solidly grounded as one would hope. And so we try to figure that out early on. And then again, we're trying to translate this promising science into novel therapeutics. And we want to engage with you as early as possible. So helps us give you some advice as well as to what experiments you might want to do or data that you might want to generate that give us more confidence. And the benefits to the PI and the university, you get equity in the new company. As I mentioned earlier, we reimburse patent costs for both background and new IP. Alleviating that responsibility from the university. There are milestone and income payments, royalty payments, and what we call reversion rights. So if a program doesn't move forward, as I said, we give back the data so that the PI you can use it any way you want. You can use it to go out, start another company, to write a grant, write papers, use it as the stepping stone for more research in your lab. And the hope is if that happens on occasion, you'll come back to us in a year or two and say, hey, I've solved this problem, or I've overcome this hurdle, or I've added new data. Are you now interested in re-engaging? And the short answer would be, in most cases, yes, we would be. We don't see this as a one and done with any one faculty. We want to build this continual dialogue. And as Eva said, if you have an early stage idea and you're not sure if it's a good fit, please contact us. So the next slide just sort of wraps things up. There are a few other slides that are, I'm sure will be made available to you that just give some background on what's available at Evotec. But with this, I'll stop here and be happy to, with Donnie and Eva, would be happy to answer any questions you have.
Tom, maybe first start off with the one that's in the queue.
Sure. Yeah, so identify a leading compound. Can you help us modify it? No, or do I need a chemist? The answer is no, you don't need a chemist. That's one of the things we offer. I've had a lot of great conversations with Mike Jung and Patrick Haran. You have some good medicinal chemists there, but what we can provide is a team of medicinal chemists doing nothing but lead optimization on your molecule. So that's absolutely within scope of what we would offer.
There's another question which when I typed the answer, moved on to answered question. Arun Singh, what are your thoughts about developing drugs for relatively rare oncologic indications in areas where there aren't good agents? Happy, Tom, Donnie, jump in. Rare alone is not a showstopper.
Yeah.
The main questions are, what are the reasons for not having therapeutic agents? Perhaps the pathophysiology is not known. There's no clear clinical endpoints, no regulatory guidance. We kind of have to parse out what are the reasons for not having therapeutic agents there one by one. So I'm not sure if anyone wants to give an example, but rare alone is not an issue. In fact, it's easier to develop a drug there if there's the right conditions.
Yeah, and we didn't mention that. We should have added that to one of the slides about what was in scope. Rare diseases are fine. We're okay with that. I mean, some are very, very rare and it's just difficult to do clinical trials, but what's considered by the FDA a rare disease is not a challenge. But as Eva said, you wonder why there are no therapeutics in that area. There are some areas that are just extremely difficult. Some are rare, some aren't. I mean, everyone would love to have a molecule to treat Alzheimer's disease, but it's just a really, really hard indication. May require multiple drugs working in concert, or we just haven't found the right single target yet. But we're fine with that, and we're not at all. Oncology is an area of good partnering opportunities. Every cancer is different, and so it's not one drug treats all. And I would say in that case, we'd be happy to talk to you about the project that you have.
Yeah, I would just add that, actually, and then Tom alluded to it, the definition of rare in the US is fewer than 200,000 patients, right? And that's almost all oncology indications, right? There's actually very few that are more than that. Yeah. So it really is definitely on the table.
Yeah. There was a question emailed to me from a colleague who wasn't able to participate today. I think it's for Eva, and it's whether you have capabilities in ADC and BiTE technologies.
That's an interesting question. Manufacturing, yes. I have to, see more specifically on developability, manufacturability for sure. I think oftentimes, the innovation comes from either what it targets, what combination it targets, but certainly we have the ability to find the right molecule, design the right molecule, and make the right molecule, including the chemistry to bring it all together.
Yeah, and I can tell you the folks at Just Biotherapeutics, a company that Evotec bought a little over a year ago, are a lot of ex-Amgen, Immunex folks up in Seattle. They're superb. So they can optimize manufacturing. They're very, very good. And Evotec itself has a very good biologics group to do the early discovery work. Been very impressed with them. There's two questions. One, if a new company is formed, does the PI have a seat on the board? I'm not referring to the SAB, rather the board of the company. That's a decision that'll be made by the management team and the investors in the NewCo. It's not a question that Autobahn could answer because it wouldn't be a decision that we would be involved in. And I think it would. I think the short answer is it would depend. And then if IP is generated after the partnership is formed, who owns the new IP? Michelle, do you want to take a crack at that? Michelle?
I'm not sure the participants can they speak, or is that allowed?
Oh, that may be the challenge.
Technically.
I can allow them to talk. Was it Michelle? There she goes. Let me
yeah.
here. Okay. I think she's been Promote her to panelists.
Oh, she's not on the call, she said. So,
Oh, okay.
okay.
So it really depends on, right, whether the IP is jointly generated under the collaboration, right?
Yeah.
If you're referring to joint IP that we have generated under the collaboration on a particular, encompassing target efforts that we're moving towards IND, then, that will be folded under the collaboration. If there's I'm just hypothesizing here, if there's another set of data that is not related to the same target, but can somehow address the same disease, the same project, maybe similar pathway, that certainly is a separate IP.
Hi.
I don't know where the question went. Let me see if I answer.
Your answer. Michelle's on now. Michelle?
Yes, I am. Hi, thank you. Hopefully my headphones will last long enough to answer the question. So with respect to new IP generated after the partnership is formed, it follows inventorship. Ownership always follows inventorship. And so if it's jointly, invented new IP that is jointly owned by UCLA as well as Audubon. And just quickly going back to, somebody had a question with respect to, does the founder have a seat on the board of the program co? So again, this is the company that's formed initially when, we identify a project and want to start. The answer to that question is actually no. And so with respect to the question that Tom was answering at the Series A, that is completely dependent upon the management and the details of that financing. So hopefully that answers both of those questions in one fell swoop.
Good, thanks, Michelle. Any other questions? Well, again, I want to thank everyone for joining and participating. I encourage you to reach out to us if you have a project or an idea for a project that you think might be a fit. We'd love to talk to you. You can also reach out to Mark and Judy and the BDOs in the Tech Transfer Office in TDG to get more information as well. As we've said earlier, we pre-negotiated, Mark said we pre-negotiated these terms, so we can move very quickly, and you can go to them to learn more about that. But what we really want to do is encourage you to think about ideas you might have, but haven't followed up, at least on the commercialization side, to develop a therapeutic, because it's not really your lab's focus, your expertise, or your interest. But it might be a nice collaborative project that would benefit patients in the long run. We're happy to suss those out with you, vet the opportunity, and give you our feedback. So with that,
thank you so much for a great and very informative panel today.
Yes.
We really appreciate it. And as you said, Mark and I are available if there's questions that we can answer. So thank you all very much.
Good. Thanks, everyone. Appreciate it. Stay in touch or get in touch. Bye-bye.
Bye.